We are always one step away from the clinic. For my research and for other research on brain tumors, we have people in my lab who are literally in the clinic every week, running samples back and forth, in the operating room, talking to our surgeons. There's so much across that bench to bedside and back, and I think that's what needs to happen for us to make progress. Bench to bedside or bedside to bench can really go both ways. You find something in the lab and you figure out how to bring that to the bedside.
My research relates to developing new treatments for brain cancer. So we decided to use the immune system of the patient to get rid of any remaining tumor cells that the neurosurgeons can't see, because you can take the mass, but you can't take a single cell right from far away in the brain. And then that cell gives rise to a new tumor, and that's what you call a recurrence.
So what we decided to do, we were working on gene therapy, and we decided to harness the power of the gene therapy vectors to modify the internal immune environment from the brain tumors.
The metaphor that we came up with was putting a brain in the middle of a blank page, right? And then in the center of the brain, we put a leaf from a tree with a lot of eggs inside it. And those eggs represent the brain cancer. So we did a life cycle of the monarch butterfly, and that in a way represents the life cycle of the brain cancer, how it turns out from being a deadly cancer, and through our work, the gene therapy will reprogram the ecosystem of the cancer to make it suitable so that the immune cells can really kill those cancer cells. So we use our gene therapy to turn those eggs into a beautiful butterfly that leaves the brain, flies away, and the brain turns out to be a healthy brain.
I study specifically a type of brain tumor called pediatric diffuse midline glioma, or DIPG. I study it both in the clinic and in the lab. So on Wednesdays, I see patients with this tumor, and the rest of the week I'm in the lab where we're studying models of the tumor to try to learn and come up with better therapies for the patients that we see. You might imagine a situation where a physician is talking to a family member and a question comes up from the family, and that question has not been answered in the lab. Really, the projects that I like to be involved with are really just like continuous back and forth, where if you're trying something you found in the lab, you're trying it in that patient, and then you're actually extracting samples. Those are going back to the lab to learn in real time, how is that patient responding? Our lab was interested in how ONC201 was specifically working in these diffuse midline glioma cells. And it is an interesting drug for our patient population. Not only is bioavailable, so it can be taken by mouth and get into the bloodstream, and then beyond that, it can get from the bloodstream into the brain. It crosses the brain barrier. So now that the drug is approved, it's prescribable. The approval is for patients who have recurrence. We see evidence that the treatment is helping slow the tumor. It does give my patients some more flexibility and opportunities to access a promising drug that wasn't there before. That's probably the most important thing.
You want solutions, and it's very hard to understand that research is slow, that research has many failures, that you have to try many things. It took us 25 years to develop this viral vector gene therapy. And if you have a patient that has a brain cancer and the prognosis is a year and a half, they can't wait. So I think that's where the push comes.
We completed recently a phase 1 clinical trial on 18 patients, and the results overall were very encouraging. compared to historical controls, when the survival at 2 years is only 5%, at 2 years we had a survival of close to 30%. That looks encouraging, and we are now continuing trial where we are combining our approach with other approaches that stimulate other parts of the immune response.
If you could just imagine, for your own child or for, a family friend who has a young patient, if they were given a diagnosis where they had a prognosis of 1 to 2 years, how urgent would we need to come up with something to make those treatments better?
We tend to work on the weekends. Basically, the patients have no breaks. Patients don't get a weekend off. That's how I see the work we do, that we owe them to try to move this forward. I like to think that it's not that we don't have a treatment yet, but we can visualize it and hopefully make a difference in patients' lives.